Hello everyone, Dr. Young here, and in this video I'm going to introduce you to enzymes. So now enzymes, right, like you may have seen in the previous video when we were talking about kinetics, enzymes are catalysts that are proteins, right? So that's really important to remember, right, that they're catalysts and they're proteins, specifically, right, which is another way to say polypeptides. And you might recognize from this enzyme over here on the right-hand side, we've got an enzyme that looks like it's got sort of a really nice symmetry here, but this is catalase, where you remember that we got the primary sequence, which is just the amino acid sequence itself. We have the secondary structure, which is what describes things like these alpha helices and these beta pleated sheets. The tertiary structure describes, right, like how do these fold together, why do they fold that certain way, and then quaternary would be if some proteins had multiple subunits. I'm not sure that this one does, although it looks like there are two kind of things working in together. I'm not sure if they're separate peptides. But anyways, right, they're proteins, and so everything that we know about proteins is true about enzymes. And remember that we said that since they're catalysts, the idea here is that catalysts are going to lower the activation energy, right? So we're going to lower the activation energy. And so if I look at this energy diagram here, right, if I'm talking about the forward reaction, I'm talking about turning A into B, the idea is that a catalyst might do something where it lowers the activation energy of this step here. It provides a new pathway so I can turn one thing into another. And so now my new activation energy for the forward reaction would just be this, right? So now I'd have an activation energy for the forward reaction for A turning into B that has been lowered. But what is important to remember here is that it also catalyzes the backwards reaction, which is something that people forget about a lot. So if I'm talking about the back reaction going from B to A, right, from B to A, it also, this new pathway, that what I drew in purple here, it also lowers the activation energy for the back reaction. So it lowers that E sub A also. Where for B, the original activation energy would have been all the way up to the black line. If the catalyst is added, it only goes up to the purple line. So you also lower the activation energy for the back reaction. So it catalyzes both the forward and the back reactions. Enzymes, no catalyst in general, but enzymes don't know forwards from backwards. They just know that they're providing a new pathway for a reaction. So they don't change equilibria. They don't change the starting or ending energy values. They just change the size of the hill separating two intermediates or two products and reactants. And then generally, like all catalysts, right, they're not really consumed in a reaction. So one enzyme could catalyze hundreds, thousands, tens of thousands of reactions before something goes wrong with it and it starts to fall apart and has to be broken down for its amino acids and then new protein, a new enzyme made in its place. And there are tens of thousands of enzymes known, so many, partially because they're so specific and we have so many reactions that we need to do, which we'll talk about more here in a second. So now, one thing, when we talk about enzymes, when people hear about enzymes, normally people think about digestion, right? They're like, oh, digestive enzymes, and people try to sell you digestive enzymes and tell you not to cook your food so that you have better digestion and all those sorts of things. But there are lots and lots of different types of reactions. A number that's thrown around is that there's about 5,000 different reactions that enzymes catalyze, right? So thousands and thousands of ones. It's not just stuff for eating. Yes, there are examples that involve food. So like salivary amylase, right, takes amylose, right? So amylose, which is a component of starch, right? Amylose and amylopectin are what make up starch, and helps break it down into glucose. And then it's called salivary because that's what happens in your salivary glands in your mouth. So as soon as you eat something high in starch, the idea is that you secrete this salivary amylase and it starts breaking it up and turning it into glucose, which is why if you take like a piece of bread and you just chew it for a while, it starts to get sweeter and sweeter and sweeter because more glucose starts to get made. Similarly, if you take DNA polymerase, right, this DNA polymerase would take DNA and help you depolymerize and repolymerize DNA to and from its nucleotides. Right, your A, T, C, G, U, all that sort of stuff. And notice I'm drawing a back and forth arrows here because, again, enzymes don't catalyze just a forward reaction or the back reaction. They do both. So sometimes, like DNA polymerase makes it sound like it's only for polymerizing, but it's also for depolymerizing. It works both ways. Alcohol dehydrogenase you may have heard of, right? When people talk about alcohol, ethanol. Alcohol dehydrogenase is what your liver produces so that you can turn your alcohol, your ethanol, into acetaldehyde, for example, which eventually gets further oxidized into acetic acid. So this would be an example of an enzyme that helps to oxidize things, in this case ethanol, but again, it goes back and forth and back and forth. Just one more example, catalase, super important enzyme for anything that's around oxygen. Oxygen, absolutely required for life. We really, really need it. However, it also does a lot of damage to us, right? It does a lot of oxidative damage. You might have heard that we're supposed to eat antioxidants, and that's so we can combat the effects of the oxygen that we breathe. We don't want to burn too fast. We want to burn just right, so we need to kind of tamp down all of the reactions that oxygen does. And one of the things that oxygen produces is hydrogen peroxide. So hydrogen peroxide, which is H2O2. And so what catalase does is it helps takes this reactive oxygen species, hydrogen peroxide, right? You may have used it on a cut or something. It stings, it kind of bubbles. We know it's pretty reactive, it's not stable. And it helps turn that hydrogen peroxide into nice benign water and then just molecular oxygen. So just the stuff in the air, and I'll balance this equation because it bugs me if I don't. But catalase helps turn hydrogen peroxide into just water and oxygen. And you can see catalase work. It's pretty fun if you just take hydrogen peroxide from the store. You can either dump in yeast and you'll see their catalase make the peroxide start to decompose and you'll see oxygen bubbles form. Or sometimes people use just potatoes. You can freshly cut a potato, pour hydrogen peroxide on it and see it bubble. That's the catalase working to make your oxygen gas. And so you'll see bubbles and stuff like that. You may have in a biology class used a cow liver or something. There's all these kinds of ways to see this. But the point is enzymes have lots of different functions, not just digestion. They help catalyze all kinds of reactions in the body. One thing I wanted to point out here, right, is you'll notice at the end of all of these names, they all end in ase, right? So we have salivary amylase, DNA polymerase, alcohol dehydrogenase, and catalase. Most enzymes, not all, but most enzymes tend to end in ase. So I'll put a little note here. Enzyme names tend to end in ase. So ASE, tend to end in ACE. I'll talk about an enzyme here in a second called like pepsin, for example, which doesn't end in ACE. But again, most of them do. So let's talk about their specificity, right? One of the reasons why we have so many enzymes is because enzymes tend to be specific, not promiscuous. And by specific, what we mean here is that they tend to work on just one molecule or one class of molecules. So by specific, we mean that they only work with one, or I'll put parenthetically, or one type of molecule. Or one type of molecule is the idea, right? And so what that means is that a given enzyme, and I'll do some examples in a second, only works with like one type of thing, which is why we need to have many. And the reason for this, it's because enzymes have a certain shape. It's because enzymes have a very specific shape. If you remember back to our discussion about proteins, right, we talked about that the shape of a protein is really important and that determines its function. Since enzymes are proteins, that's equally true, if not more true here. And so if you take a look at these two diagrams on the right-hand side here, we have what's called the active site. That's what's here in blue. And the active site is where our substrate, right, our reactants here, so here's our substrates, which is what another name for reactants when you talk about enzymes. So these are our reactants. Whatever is going to get changed by this enzyme. Your substrates are drawn right here in these little, I got a hexagon, a little rectangle, but then there's actual molecules drawn here. The active site is where those fit into the protein. So I got little blue kind of crescents here, versus I've got the stained blue pictures over here on the actual enzyme picture itself. This is a type of kinase, which we'll talk about in a second, means you're adding or removing a phosphate. So the idea is that because they have a very specific shape, that it has to match, right? It's like a lock and key. Sometimes it's called a lock and key mechanism where, right, the protein itself is kind of like the lock, and then the substrates, the reactants coming in are like the key. They have to be the right shape in order to work, right? It's kind of like the idea of like Cinderella and her glass slipper, or the idea why, you know, I can kind of tell when I have the shoes on the wrong feet, right? Things need to fit the right way, right? You can't put your glove on your foot and have it fit real well. They're different things. Your foot's a different shape than your glove is, and so it doesn't quite work. And it's the same idea on a molecular level for substrates and their enzymes. So most enzymes are pretty specific because they have a certain shape. They need to fit in this active site for the actual enzyme to work. If they're not the right shape, they don't fit, and so the enzyme doesn't work for that. And just as a side note, as promiscuous, some enzymes are a little promiscuous, but very, very few. Promiscuous just means it works on a large range of substrates. So just say works. Let me erase that. Works on a lot of substrates. As a side note, it's a goal of the biotech industry to take enzymes and tweak their structures to replace certain amino acids with other ones to make them more promiscuous. Because if you can make enzymes that work with lots of different substrates, then you can use them in industry to help catalyze and make reactions for a lot cheaper with a lot less waste. So making promiscuous enzymes is kind of a good thing. But in the body, our enzymes are very specific. They do one thing. So like, for example, right, so salivary amylase I was talking about before, right, that's what helps break amylose into glucose. So if I have amylase, right, again, ACE versus OS, if I have amylase, that helps you break down amylose into glucose. That's what we want. However, if I had cellulose, right, which is fiber, has a different type of linkage, if I try to add amylase to cellulose, I don't get anything, right? I don't get glucose. I have no reaction occur because it's not the right thing, right? Remember that amylose had the alpha 1,4 linkage and cellulose had the beta 1,4 linkage. They're really different shapes, and so you can't expect the same enzyme to work for both. If I did want to break down cellulose, we would use cellulase. Cellulase is a different enzyme, but like the name suggests, it helps break down cellulose, and then you would get your glucose, and that would work. And so again, the idea is that the enzymes are very specific. We've probably all heard of lactose or lactase, right? Some people are lactose intolerant. Lactose is a sugar in mammal milk, and lactase is the enzyme that helps break it down. And so if you take your lactose, you're going to turn it into glucose and galactose, and that works great. Now, some people have stopped expressing lactase because normally mammals wean and they stop drinking milk, and so the body just stops producing lactase. Why bother making an enzyme that you're not using? But in some cultures, right, we still drink milk, and so we keep on producing lactase, so we keep on being able to break down lactose. So we can still drink milk along into our adulthoods, which is sort of odd for mammals in general, because, again, normally mammals wean. But when people stop producing lactase, then they can't break down lactose. It goes to their gut, and microbes end up having to metabolize it, and then you get gassy cramps, and all kinds of bad stuff happens. But again, lactase is very specific. It works for lactose, but not, for example, sucrose, right? If you have lactase trying to react with sucrose, you're not going to get these things. It's not going to work. Again, you're going to have no reaction, right? Because lactase is specific for breaking down lactose. Right, if you want to break down sucrose, you probably have an idea what you'd use. It's just sucrase. Right, so you would need sucrase to break up sucrose, and then you would get your glucose. Oh, sorry, I should have changed that. Shouldn't have said galactose for those. It would be fructose. That's what I get for copying and pasting. So fructose, not galactose. Right, because sucrose is a combination of fructose and glucose, not glucose and galactose. My apologies. So again, just the idea here is enzymes are very specific. They only work on the thing they're supposed to work on. Now, another thing to keep in mind is that enzymes are sensitive to their environment, that their activity can be affected by things like temperature, like pH, and like salinity, right? And by salinity, I mean the amount of like salts, right? The amount of solutes. So I'll put solutes here. And I cannot work this today. That's fine. Solutes. There we go. Solutes. So I have an example here of a graph showing the relationship between activity and pH. But I want to mention, right, if you go back and remember about proteins, since enzymes are proteins and proteins can be denatured, that means that the things like temperature and pH and salinity will affect the enzyme activity, right? Because if you denature a protein, or in other words, you mess up that tertiary structure, right? So interfere with the secondary or tertiary structure, then you will change the enzyme shape and therefore its substrate, therefore its active site and its ability to do whatever. So here's some examples of pepsin and chymotrypsin. Again, this is looking at pH. So we have pH here. So let's say this is a pH of about 2. Let's say this is about 10, and then we have 7 in the middle. And then percent activity, this is talking about rates. So this would basically be no reaction down here. So that would be like no reaction. And this would be the fastest reactions up here. 100% would be the fastest it can go. 0 would be that there's just no reaction whatsoever. And so if you take a digestive enzyme like pepsin, right, these are both enzymes that work on proteins. These are both proteases. So it breaks down proteins. Put breaks down proteins. And the idea is that pepsin breaks down proteins, and pepsin works in your stomach. So it gets excreted into your stomach when you eat things like protein. And you'll notice that pepsin seems to work really well at a low pH of about 2, which happens to be about the pH of your stomach. Your stomach's usually between 1 and 2, depending on what you're eating and depending on the person. And so pepsin works really well in your stomach, which is great. That's where it's acidic. As soon as your digestive fluids, everything that's in that bolus, that digestive bolus, all of that, juice and all that stuff that you've been eating moves from your stomach into your small intestine, the pH goes up. And so as the pH goes up, as it starts getting closer to like 6 or 7, the idea is that pepsin's activity decreases, right? Now it only goes, now pepsin works far more slowly than it did at a low pH. So pepsin sort of gets deactivated. But what's kind of nice about this is that that's when chymotrypsin gets added to your small intestines, and it's another type of it's another type of a protease. Also breaks down proteins, but works in a different way. Breaks down proteins also. Only this is in the small intestine. And so these different enzymes have different functionality at different pHs, right? Chymotrypsin works the best at about 7-ish, whereas pepsin works the best at about 2-ish. And so they can be activated and then denatured depending on the pH of their surroundings. It's kind of a good thing, for example, that pepsin doesn't work very well at a pH of about 7 because it's a really powerful protease. It breaks down proteins really well. And so if pepsin gets out of the stomach, it can really destroy a lot of the tissues around you, right, since you're mostly made out of proteins. And so once it gets out of the stomach, you don't want pepsin to work anymore, right? Your pH of the surrounding fluids is about 7, so once pepsin leaves your stomach, it hits a pH of 7 and it gets deactivated, which is real great if you have like an ulcer or something like that. You don't digest yourself from the inside out. But the point is that enzymes are very sensitive to their environment, whether it's temperature, pH, salinity. There's all kinds of different things you can do to affect the activity of an enzyme. Now, there are different types of enzymes. Like I said, there are 60, 70, 80,000 different types of enzymes. But I want you, and I don't care that you guys memorize them. I don't care that you know like what a fructophosphokinase is or something like that. What I care about is you have a sense of some of these major classes of enzymes. And for my students, this is the list of the 5 major types that I want you to know about. There are more than this. There are like transferases, for example, decarboxylases. I don't really care about those as much. Again, this is for my students. But I do want you to know these 5 because we're going to use these 5 to talk about the cellular respiration and glycolysis and the Krebs cycle, etc. So I want you to know isomerases, which just catalyzes the isomerizations, right? It just sounds like the name. So this would be like, for example, turning glucose into fructose and vice versa, right? Those are just isomers, right? They're both C6H12O6. They're isomers of one another. Oxidoreductase, that catalyzes oxidation and reduction reactions, redox reactions. So that's like adding and removing oxygens. So that was kind of like what I showed you with that alcohol dehydrogenase. That's a type of oxidoreductase where you're increasing the number of bonds to oxygen and therefore oxidizing something. And so we're talking about that's that type of reaction. So where you're doing oxidations and reductions, and I'll have more videos about redox reactions here posted soon. Now, hydrolases, those catalyze the hydrolysis and condensation reactions. So, all right, so that'd be something like taking two alcohols and doing your condensation reaction to make them, in this case, an ether plus water. Or again, the backwards reaction, right? The hydrolysis of that, taking that ether and cutting it to make the two alcohols. Now, it's not like your cell makes diethyl ether or anything, but this is just the type of reaction, right? So hydrolysis and condensation, like we talked about before. Now, hydratase sounds a lot like it, but hydratase is the addition or removal of water from a molecule. So it's not adding water to like break up a molecule to split it into subunits. It's just straight up adding it usually to like an alkene, for example. So it'd be like if you had water and an alkene, this type of enzyme would catalyze the addition of that water. And let me do this in blue just to kind of emphasize it. The addition of water to that molecule where you add an OH and an H. And that's what came from this water molecule here. I'll erase this and draw it in blue. Right, that's where this water went, is it was added to the molecule. You've added the element of water, or again, removing it. So these would be hydration and dehydration types of reactions. And then lastly, kinases, which catalyze the addition or removal of phosphate from the molecule. Right, so if I had, again, some alcohol, we're going to see this pretty much exclusively with ATP, right? Remember that ATP, one of its two main jobs was to phosphorylate things. And so we'll see ATP come along to help add a phosphate onto like things like an alcohol. So that's an O up here, which means it turns into ADP. And so we'll see these types of reactions coming up too. Also super important for not only metabolism, but also just signaling and activating proteins and stuff like that. So the kinases, I think, sound the funniest because kinase doesn't sound anything like phosphate. But all these other ones kind of sound like what they do, right? Isomerase helps isomerize things. Oxidoreductase is involved with oxidations and reductions. Hydrolase, I always think about this in terms of like hydrolase. I feel like hydrol sounds like hydrolysis. So I think hydrolase and hydrolysis sound a lot alike. Remember, that's the one for that one. Versus hydratase is different. Hydratase is for like a hydration. So hydratase is the other one. They both sound like water, but it's different. And then lastly, kinase. So key things to remember, right? Just enzymes lower the activation energy. They catalyze the forward and the back reaction. They catalyze many different types of reactions. It's not just about digestion or anything like that. They get consumed. They don't get consumed in reaction. They can usually catalyze tens of thousands of different reactions. They tend to be specific. They tend to be affected by their environment. And I want my students to know those 5 main classes of enzymes because we're going to apply those in future videos. So hopefully you have a little better idea of what enzymes are and kind of how they work. Keep an eye out and we'll apply these different classes of enzymes in the future when we talk about glycolysis and the citric acid cycle and cellular respiration in general. If you have any questions or comments, please leave them down below. Happy studying.